Edu Arctic2, Polar Research for Education, innovative program in Poland and Norway. Webinars.
Welcome everyone to this webinar about microbiota and the microbiome, in which I will start to explain a bit how we're using microorganisms in our research and what we want to know about them. My name is Cornelia Klutsch. I'm a research scientist at a biological station called Nebius Swanhoft. And before we start, I thought I'd introduce myself a bit more. And I work at a station that is here really far in the north of Norway, directly at the border to Russia and very close to the border of Finland here. On the left-hand side, I just included a few photos of the station itself in the summer. Right now it's very snowy outside still. And up here, you see the station here and the river, the Pacific River, which is one of our main research areas we have here at the station. And we really can look to Russia and greet our Russian friends on the other side. So it's really close to the Russian border. And the station has a portfolio. We research several different things. And here I just listed a few. Traditionally, there was a lot of brown bear research, but in recent years, we have really expanded in different directions and have a lot of different species and research projects on those species at the station. And this includes also a lot of fish research that are important for the local communities. And I will talk a bit about microorganisms and fish health and a few of those research projects we are conducting or we are planning to do in the future. So what I would like to discuss today is, first of all, I will give you a few definitions of terms I will use throughout the talk so that you're a bit familiar with those. And this is really the base to understand then the subsequent slides. But also, why are we so interested in bacteria and viruses and other microbes? What is it that can the study of those organisms give us for our research? And then I will talk a bit about different concepts a bit and why this is important. And then I give you a few examples from our research, specifically the fish microbiome and why this is important. And then I will expand a bit in the end to a few other interesting areas and areas where research is really active. And why this is important for us humans, actually for our human well-being and our food security, for example. And in the very end, I hope I have time, or we have time, to have a little quiz with Kahoot. So I'm announcing this already now so that you can probably get your devices out, your mobile phones and so on, so that you're ready at the end of the presentation. And finally, I would just like to mention that we have also a chat. So if something happens through the presentation, I don't know, the sound cuts out or something happens, you can notify us. And also, if you have any questions regarding the presentation, please feel free to just comment or post your questions in the chat. So I will probably answer them then at the end of the presentation. We'll see how it goes, but so we can collect some of those questions probably. Okay, so then let's get started. So first of all, I would like to explain a few terms. Microbiota is really the entirety of all microorganisms. This includes bacteria, viruses, protists, and fungi. Fungi are basically mushrooms, just very small, at the microscopic scale. And then we talk a lot about biomes in this presentation. And the biome is an ecosystem consisting of flora and fauna, usually at a large scale, what we can see. And this leads us to the term microbiome. And this is really the merger of these two terms, microbiota and biome, a microbiome. And this is something very small, microscopic, small, but that consists of different species. and is an own entity in nature. This is then the collection of all microbiota of all these different species of bacteria and viruses and so on, and their genes. This will become important, their genetics, in a specific place. For example, as here shown on the right-hand side, the gut microbiome. So this is really a term we are using to describe the entirety of those organisms, of those microscopic organisms in the intestine, for example. But there are others. It's just an example that we use here. And just to drive home already a bit the importance of those microorganisms, I have collected a few fun facts, just to give you a sense of the dimensions we are talking about. So first of all, 50% of your body consists actually of cells, bacterial cells and viruses and fungi cells. So 50% of you is actually consists of microbiota. And just in the gut, in the intestine alone, we carry about 5,000 different species of those microorganisms. So the human body itself is actually an ecosystem, and we carry a lot of different species in us. And the entirety of the microbiome would probably weigh about 2 kilograms. And then there's another very interesting fact for us researchers. And this is that all of these bacteria have their own genomes, their own genetic material, their genes. And the entirety is probably about 200 million genes, so a very, very large number of genes. And especially if you consider that the human genome has probably about 20,000 described genes. So there's a large collection of genetic material in us, in those microorganisms, that has been also basically termed a second genome. So basically, there's a lot of material that can have certain functions. And we'll talk a bit about this in a minute. But before we go there, it's probably interesting to think about why we are studying microorganisms now in so much detail. And this is really going back to a technical advancement in recent years. So around 1600, the first microscope was developed. And then in the mid-1600s, the first microorganisms were described. And then about 150, 200 years later, there was a connection made between those microorganisms, mainly bacteria, and disease. And this is probably the reason why microorganisms have a very bad reputation, because we mainly connect them to disease. But this is actually not true. I will tell you and explain you a bit later why microbes are so important to us. And from that time, there were techniques developed so that we can grow a lot of these microorganisms in the lab, and we can describe them with the help of a microscope. However, those microbes that we can culture or grow in the lab is actually only a very small fraction of the entirety of species we have out there. Most of the species we can't actually grow or culture in the lab. What has happened then since the 1990s is that we use genetic methods, DNA sequencing, to actually detect those other species we cannot grow and see under the microscope. So this technical development that we are now able to sequence genes from organisms at a large scale has enabled us to study microorganisms in far more detail than in the past. And what we are doing, for example, here at the station is that we collect either feces, or if we have deceased individuals, probably the entire intestine. That is then we have everything in a tube that we can carry to the lab and do some analysis. And of course, this tube contains and also the microbes and their DNA. And then the lab, we can do some techniques that in the end we actually can sequence those different microbic species. What this leads to that we get sequences of the 4 nucleotides that are in the genome, A, T, C, and G. And I'll just give you one example here, a very little example, and I have highlighted differences between different samples, for example, so we can identify differences between them, and then we can identify, for example, different strains or different species of bacteria. And at a large scale, this will give us then information about different composition in different environments, where the fish live, or if they have a different diet, for example. And I will talk about this also in a bit more detail. And what has happened in recent years is that because we can sequence more and more, we can get more information about those genes that are involved, is that a certain concept has developed called the Holobiont concept. And what this means is that we get more and more knowledge that, for example, our microbiome, the human microbiome, has effects on our well-being, on our immune system. So we have a host genome, either, let's say, from a human, but there can be others in nature, like a tree. Trees have also their own genome, which is called the host genome. So they have their own genetic material. But the microorganisms that live in or on those organisms, let's say humans, have partially co-evolved with us, and they have different functions. And their genes have different functions and can affect the host, let's say, human immune system, diet, nutrition acquisition, and a lot of other things. And because there's this dependency between the microbiome we carry and our own genome, this has led to the concept that this is an entity which is called the Holobiont. And it's important to realize that there are other microorganisms that have nothing to do with us in the environment. So there are additional microorganisms out there in the environment, but they have no connection to us directly. And what researchers have done in recent years is to test this concept. In the beginning, it was just an idea that there might be a connection between the genomes, the genetic material in our microbiome and our own genome. But then they tested this idea, and this is what really scientists do. We're trying to come up with hypotheses that we are trying to test. And one group tested then specific fish species, and they grew different specimens in colder temperatures and in warmer temperatures. And what they wanted to know is if this is true, that the Holobiont concept exists. So you have this dependency between host genome and microbiome and their genes. Then we would expect to see a difference in the composition. So which species are, for example, associated with these two different groups. So the temperature resistant one, which would be the fish that can survive in colder temperatures, for example, and the temperature sensitive fish type that needs warmer temperature and is more sensitive to colder temperatures. And setting up this experiment, scientists could show that indeed these two different groups that were in different environments had different microbiotas, different species of microorganisms in them. So, which means this Holobiont concept seems to be true, that we have specific composition of microorganisms depending, for example, in which environments organisms live. And this is, of course, very interesting for us. For example, we are living up here in the Arctic, 400 kilometers north of the polar circle. And the conditions here are very, very different than, for example, in Italy or further south. So there are probably very different interactions between fish that we have here and their microbiomes than in other places. And to summarize this a bit more generally, we have different factors that can change the number of microorganisms organisms carry and the species, which species are associated with different conditions. And the other thing is that the combination then of those species and the abundance determines also a lot of things and have certain effects then on the host. So for example, we have what we call intrinsic factors. So these are factors from the organism itself. So you have a fish species, and the genetic background of this fish species and specimen will determine which microbiome it will have. Then there are other factors. Age, reproductive stage, and sex can lead to differences in the number and the number of species in the bacterial composition. Then stress, if there's disease, and how the immune system functions can determine differences. And then you have the nutritional status, if the specimen is well fed or not, if it's starving. And also this can affect the composition of microorganisms. And then we have so-called extrinsic factors, and these are simply external factors outside of the body of the organism, like environmental conditions, like we said, cold and warmer conditions, for example, temperature, light exposure. And fish, specifically probably the water quality, if we have clean water or polluted water, and the diet that is ingested. And all this leads then to specific differences in the microbiome that then determines other things, like, for example, how well an organism can digest food and extract nutrients from food. It can determine growth. It can determine brain functions. There's research coming out that microbiomes are connected to memory, for example. Then it can have effects on the immune system and reproduction. So what this means is that our little friends in our intestine, for example, or in other places, these microorganisms have a very, very important function, and their genes they carry can lead to specific differences. And I thought I will just summarize this and repeat this one more time in a bit different way to really drive this point home. So microbes have these very, very important functions. For example, that environmental factors, what we call also abiotic factors, that you have, for example, stress because there's something happening in the environment, the temperature changes. If you have a high diversity in your microbiome, you may have a higher tolerance to this type of stress. Then, of course, there's a protection against pathogens, and connected to this healthy microbiome improves the host immunity system. Then the host genotype, this is this Holobiont concept again we discussed. And growth and development is very dependent on microbiomes, as well as nutrient acquisition and nutrient extraction. And just to drive this home, there's a very close connection to host environment interaction because of microbiomes. And I told you in the beginning that we will talk a bit about fish and what we are doing and why this connects also to food security. So fish are very, very important nutrient resource for humans. And we cultivate, we grow fish and breed fish in fish farms and in hatcheries, also for conservation purposes, not here at the station, but I mean in general. And some of these fish are released into the wild again for supportive programs to help natural population survive and thrive. If, for example, the population can't reproduce on its own enough to build enough offspring. So what happens in these agricultural facilities is that, and this is also very recent research, that the microbiome becomes very, very important, because if we're releasing fish, we want to have healthy fish out there. However, because in rearing facilities, you have fairly small enclosures, you have a high density of fish. What happens is that you can have more infections of the fish. So the microbiome is very important to have a good immune system. But often what we find in agricultural facilities that they have also specific treatment. For example, antibiotics are added to the water. And the problem with this is, of course, that antibiotics are targeting bacteria, mainly bacteria, and this changes the composition of the fish microbiome. Then you have other aquaculture practices like the diet, for example. In aquaculture, you have a lot of sort of fish pellets that are used, and this is not the natural food the fish would eat in the wild. And also this can change, then have an effect on the microbiome. And then, of course, factors like water quality, how much salt content do you have, because different fish species are, of course, used in the wild to different conditions. And all of these factors can then affect the specimen's microbiome negatively. So a lot of research is actually going into this to optimize the conditions in these agricultural facilities to rear healthier fish. And if they're released into the wild as supportive practices, then of course the idea is that they have conditions in the facility that are very close to the outside to hopefully keep everything intact regarding the fish microbiome. So you see that a lot of thought goes into these, and these are really active research fields right now that a lot of people are interested in, to also secure food, provides food security. So we talked quite a bit about fish microbiomes and why this is important, but I think it's important also to expand a bit and just to let you know that there are other microbiomes. So for example, there are plant microbiomes, and these are usually connected to the roots of the plants. So there are a lot of microorganisms connected to that. However, more recently, there have been also specific studies that show that you have microbiomes connected to leaves, for example, or flowers. However, there are even others, and I just want to take a bit of time to introduce you to a few other microbiomes that are out there and what functions they have. And you see here a little graphic that shows diverse ecosystems like soil, the ocean, then terrestrials like land. And we start probably here at this corner here. So in soil, microorganisms are incredibly important because they are really decomposing, they're breaking down that material, let's say that wood leaves are shown here. So the function in this ecosystem is that they extract nutrients and fertilize the ground, basically. And then we have the ocean. And again, microbes contribute there by decomposing, breaking down dead materials, dead cells. However, what is probably not so well known actually is that about 50% of the oxygen is built in oceans by photosynthesis of microorganisms like microalgae and so on. So this is an incredibly important contribution to our oxygen content. And then we have terrestrial, so you have different ecosystems from arid regions like deserts to more green forests to tundra and permafrost. But what is very important here is that we have also a lot of impacts of humans by industry and agricultural and other uses, land uses by humans. And those land uses have a very important impact on microorganisms in the field, so to say. And here I just want to give you a few examples of this. So activities, human activities lead to pollution and other things that can change the composition of microorganisms in different ecosystems. This means that they can have profound effects on the ecosystem functioning. For example, here in the upper corner, we have fertilizers that are used in agriculture can go into the ground or in the river systems and the sediments and lead to more nutrients. However, this leads also to different growth of different organisms and can change the ecology of those microorganisms. Then we have land use, how we use, if we have, for example, cows and cattle and other farm animals, feeding on grasslands, for example, or if we grow different types of crops on specific fields. So how we use the soil and the land there has an effect on the composition of microorganisms. And then here on the right-hand side, we have different examples, but I just would like to mention the rice paddies here, because rice is another very important food source for humans. And about 50% of the world's population is in one way or the other fed with rice. However, these rice paddies are also known to release a lot of methane, which is one of the climate gases that leads to global warming. And also here, again, it's a land use factor that changes also microorganisms in the soils. Then we have, of course, human climate change. So rising temperatures will lead to changes. And all of these factors lead to a change in the microbial diversity. We know in diversity and abundance, like the number of specimens, but also the number of species. And usually all of these effects have a negative effect. So usually climate change reduces the number of species we find in an ecosystem. And with this, thinking about the genes again, their functional capacities and how, for example, microorganisms support plant growth. And I like really this analogy here that we talked a bit about humans. And in humans, it has been shown a lot of medical research that if you have a disturbed microbiome, that you have a low number of species, of different species, like the diversity is reduced, but also the number, then this is connected to different diseases, to stomach problems, skin conditions, and other diseases. However, If your microbiome was more diverse, you have a higher number of species and a higher number of individuals, like specimens, then you have a well-functioning microbiome, and this contributes to human well-being. And the very same can be said about ecosystems. In healthy ecosystems, we have a high diversity of microbiotic organisms, which means, as we talked about a bit, that this is important for soil, like functions in soil, like decomposition, fertilization of the soil. And this leads to higher biodiversity of other organisms, like plant species, and so on. So you have a sort of functional ecosystem. However, if this is disturbed, for example, by these anthropogenic human impacts, then you have a low diversity in microorganisms. And this leads to an accumulation, for example, of undecomposed litter. So the decomposition function of microorganisms is disturbed. This can lead to a diverse range of other problems and includes often that higher intervention, higher management by humans is important. Let's say the removal of trees or restoration effects. It means also that we have a higher risk of species coming into the ecosystem that don't belong there, called invasive species. And there are several talks, by the way, in the Edu-ARCTIC program where we talk about invasive species if we want to look further into that. And in total, this means that, for example, the ecosystem is not as resilient, as stable. And for example, if natural disasters like fires hit a specific region, that region can't recover as fast as it would with a good set of microorganisms. So I just like this analogy, how important microorganisms are for us and for the surroundings, for the environment we live in. And with this, I would just like to give you a few final notes. What I have done here is really just the tip of the iceberg. We are just trying to understand all these different interactions, all the different functions microorganisms have for us, humans, but also for our environment and the species that live in that environment. And we are basically at a stage where we are describing the diversity of microorganisms. But the understanding these interactions will take more time. And this is really the matter of future research that we're trying to find out and really understand more. And with this, I would like to thank you for your attention. And if you have any questions, then please let me know in the chat. And I will wait a bit and try to answer some, and then we can go to the Kahoot. Okay, thank you very much. So do you have any questions? You can also unmute if you want to ask questions. Are there any classes here, by the way?
How long have you been studying microbiota?
How long? We here specifically at the station started probably, thinking about this probably 2-3 years ago. And we are in the process of starting to conduct more experiments there. We have worked on fish now for about 2 or 3 years. But in general, the field has probably really started in the late 90s, but a lot of research was then focused on humans first. But really big projects, probably about the last decade, came up internationally. Any other questions?
Yeah. So why fish? Because usually, usually in laboratories, you hear about, I don't know, mice or rats and all that. And why
Yes.
fish here?
Yes, so we are interested in fish in particular because it's so important for the local community. So a lot of local people fish for their personal use, for their personal food supply, sustenance. The other reason is that in the Pasig River that I mentioned in the beginning, there are different fish species that are interesting. So for example, brown trout is a fish species that is used for recreational fishing also. So a lot of tourisms come there. And there we have, for example, an example that there's a hatchery where there's supportive breeding going on. And the supportive breeding, which I mentioned a bit in the presentation, leads to or could lead to changes in the microbiome. So we're trying to figure out, for example, if we can improve those breeding conditions. The other thing is that Norway, more generally, Norway has a lot of coastline and fish are simply a very important food item in Norway, but has also a lot of cultural importance in the country. And there's a whole industry connected to fishery and ocean in Norway. So in Norway, this has simply a high research importance.
Thank you for the answer.
Welcome. Anyone else? So you can think about questions a bit further. So what will happen next? I will try to stop sharing and then resharing my screen so that you can hopefully see my web browser.
Watch other recordings from webinars on our YouTube channel, youtube.com/EduArctic. Edu Arctic2, from polar research to scientific passion, innovative nature education in Poland and Norway receives a grant of 240,000 euros received from Iceland, Liechtenstein and Norway under EEA funds.